All right, so we left off on the last video talking about structure equals function with specifically with carbohydrates. So in our notes, we're on page 32 still. You're going to notice that you have glucose. Not only do you have glucose, which is a monomer, but you have two different forms of glucose. So here we have alpha glucose, and we have beta glucose. So take a second to look at the difference between the two. What makes alpha, alpha? What makes beta, beta? The structure is slightly different. Notice the orientation of this hydroxide ion. OH represents hydroxide. So since this hydroxide ion is oriented downward and this hydroxide is oriented upward, well, what does that mean? Structure equals function. So the function of these two molecules will be different just by that slight change. So we'll notice down here for B in this diagram, we have a starch. We have a 1 to 4 linkage of alpha glucose monomers. So if we put a lot of alpha glucose monomers together, we get starches. And in here, for this diagram, we have cellulose. We have the 1,4 linkage of beta glucose monomers. So all I really need you to take away from this is that this small change in structure resulted in a major change in the overall structure of the polysaccharide. So think about it for a second. Whenever you eat a starch, can your body break those down? Yes, they can. Your body can break down starches, but can your body break down celluloses? No, it cannot. Remember, you need help from probiotics inside of your gut. So that small change resulted in two different polysaccharides. Okay, so moving on, that is carbohydrates. Carbohydrates, remember, are your main source of energy. Your body can break them down really quick and get a lot of energy from them. Now, your next source of energy are lipids. If you're someone who does not eat carbohydrates because you want to burn fat, then what you're doing is you're breaking down your body's lipid storage. These macromolecules are fats, oils, waxes, and steroids. Most lipids are hydrophobic molecules. They contain little oxygen and are mostly nonpolar in nature. So what does that mean? Well, let's look at my little example here. So a good illustration, you've seen this before for nonpolar versus polarity is oil and water. So here at the top is canola oil that I bought from Walmart, and here is water out of the tap. So no matter what you do, if you shake this up and mix it, they will mix momentarily, but eventually the oil will rise to the top. Well, why does it rise to the top? Well, it rises to the top because it's less dense, but why don't they mix? Water is polar and oil is nonpolar. So as long as oil remains nonpolar and water remains polar, they will not mix. That's why if you get oil in your clothes, you have to get something that is nonpolar to get it out. Now, one thing that could help is using a surfactant. As you know from the water properties lab, hopefully you looked up and did a little bit of research, what causes detergents to break the surface tension of water? Surfactants in soap. So when you wash your hands with soap, you're breaking water surface tension so that the water is more effective at washing your hands. It also could help you wash off lipids, but still would not be as good as using another nonpolar substance. All right, so this is a type of lipid here, canola oil. So what causes a substance to be nonpolar? Well, remember, a polar substance has a slightly positive region at one end and a slightly negative region at another end, causing them to bond together. Nonpolar molecules do not have that. All right, moving on. Two main parts of a lipid. You have a fatty acid, which are long chains of carbons and hydrogens. Imagine just chaining together a bunch of carbons and sticking on hydrogens to the end. That is a fatty acid chain. All right, so then you have the 3-carbon glycerol molecule, which is actually an alcohol, to hold the whole molecule together. Lipids use a covalent bond called an ester linkage to hold the fatty acids and glycerol together. So what I think is a good illustration of a lipid is your hand and your finger. Okay, so your hand holds your finger onto the rest of your body. So think about for a second, the hand and the finger, what do they represent in relation to a lipid? Well, the finger is a fatty acid. The glycerol, your hand, is the glycerol. So the glycerol is a alcohol group that holds your fatty acid chain on it. Remember, the fatty acid chain is just a long chain of carbons with hydrogen stuck all around it. And what type of bond holds your finger to your hand? An ester linkage bond, which is a type of covalent bond. So is it a strong bond? It's covalent, so yeah, it's pretty strong. And now we have major types of lipids. We have triglycerols or triglycerides. These are your basic fat or oils. So if you've ever had blood drawn in a physical, one of the things that may measure are your triglycerides, your triglyceride levels. So they do that obviously because there is a healthy amount of triglycerides that should be in your blood because they do have a purpose for being there, but also you do not want too many or you do not want the incorrect ones to be there. So since everything is structure equals function, I think it's very important that we know that we draw these structures because it helps us remember what they look like. So in the case of a lipid typically has 2 main parts, as it says in our notes. It has glycerol head and a fatty acid chain. Now, if we talk about triglycerides, you can probably guess how many fatty acid chains those have. Tri means 3, so triglycerides have 3 fatty acid chains. So let's draw a glycerol head. So it's an organic molecule, so you know it's going to have quite a bit of carbon. So we have carbon linked to another carbon, linked to another carbon. You remember when we talked about carbon, one of its unique characteristics is the ability to form 4 bonds. And if you remember from chemistry, we represent a bond by drawing a straight line, like so. And the straight line represents the sharing of 2 electrons. So what are we sharing our electrons with? Well, you're going to know there's always going to be some hydrogen involved. Hydrogen is the most abundant element in the entire universe. It's very small and it's found everywhere. It fills spaces everywhere. All right. So we have 3 bonds left over here. So what is it that fills in these bonds? Well, it says in our notes that the glycerol is an alcohol, and alcohols have oxygens. Okay, so we have our oxygen. So we have our glycerol head. Now, what's next? Well, we need those fatty acid chains. So these oxygens are all obviously going to be bonded to something. So what are they bonded to? Well, that's a good guess. It's either going to be hydrogen or carbon. Hydrogen can only hold 1 bond because it's the first element on the periodic table, only has 1 electron. So it cannot be hydrogen here if we're going to form long chains of carbon. So it must be carbon. So for forming long chains of carbons, obviously the next thing that's going to be bonded here is carbon. And then carbon. All right, so we'll stop there. Now we have some spaces to fill in, right? Because carbon can hold 4 bonds. Move this out of the way. Try to. Okay, it's not going to go away. All right, so we're going to have bonds here. There we go. Bond, bond, bond. So we have 1, 2, 3, 4. Bonds all around. All right, something really important about here is that we're not going to have hydrogens that fill in the beginnings of these chains. It's actually going to be a double bond to an oxygen. Okay. Now the rest of these spaces are filled in with hydrogens. Hydrogen's just going to come in and fill in all the gaps. It's all a little too scrunched together, so make sure that you give yourself enough space. Now, the question is, how long can these, so these chains can be long, and it varies on what specifically the lipid is going to be for. You know, if you have about 22 carbons in each chain, then you have what is called a long chain fatty acid, and then you can have short chain fatty acid. But this is as far as we'll go, just to get the general idea of what a lipid is. Now understand, in between each of these bonds here, if we need energy, we can break these bonds using hydrolysis reactions and release that energy. So when your body is run out of carbohydrates because you're not feeding it enough carbohydrates, then it will resort to lipids, breaking down these bonds between these fatty acid chains. But it's not the main pathway for receiving energy. Carbohydrates are your first go-to source for receiving energy. Now, if we want to simplify this structure and make it look a little bit neater, what we can do is we can actually remove these hydrogens here. Some people hate it whenever I do one thing and then erase it and do another thing. But I need you to see this. I'm going to try not to erase the carbons that I have. Some of you probably know what I'm about to do. Fix this one and this one. So what we can actually do is just say, since we know that there will be 2 hydrogens bonded to this carbon, we can just say CH, goodness gracious, CH2. Let me just do that all at one time. How about that? CH2. And then we can have CH, how many? 3, exactly. CH2, CH3, CH2, CH3. Trying my best here with the handwriting, but it's not the easiest thing to do on this laptop. So there you go. So there is a triglyceride, 3 fatty acid chains, and these chains could be longer. And then here is your glycerol head. All right, so the last thing I want to talk about for this video is saturation. So here in your notes, it says the degree of saturation, the lack of carbon-carbon double bonds, helps to determine the structure and function of many lipids. So when you think of saturation, think of your shirt. When it's raining very heavily outside and you get your shirt soaked, totally soaked, to the point where it cannot hold any more water, it is fully saturated. Whereas if you just spray a little bit of water on your shirt, but it could absorb more, it is, maybe you would say, unsaturated. So when something's fully saturated, it cannot hold anything else. Okay, so keep that analogy in mind. You probably know where I'm going with this. And then that's the end of this video.